genetic mapping in humans. Genetic mapping is also called as linkage analysis. Linkage analysis provides the clues about the position of the genes on the chromosomes or where the genes are present on the chromosomes. This methodology is used for the identification of the disease genes. There are many single gene disorders in case of humans and in case of other animals that have been identified with the help of linkage analysis. The most common disease they are Duchenne muscular dystrophy, retinitis pigmentosa, and hereditary hearing loss. These are all single gene disorders that have been identified with the help of linkage analysis or genetic mapping. How genetic mapping or linkage analysis is performed? The researchers or the scientists, they identify the phenotype or a family where that disease is running, and they collect the blood samples or the tissue samples. From the blood samples or tissue samples, they isolate the DNA. Scientists, they isolate the DNA from the blood samples of the family members in which the disease is prevalent or it is running. Once they isolate the DNA, then they use different molecular techniques to identify for typing the DNA. Once they type the DNA, with the help of typing, they reach close to the region where there is a disease gene. Before the scientists they reach to the disease gene, with the help of genetic markers, they get an idea that where can be the disease gene or where can be the rough location of that gene. This is performed with the help of the genetic markers. There are different types of the genetic markers. Some are called as STR, short tandem repeat markers, polymorphisms, or restriction fragment length polymorphism. These are the techniques or these are the genetic markers which are used to reach close to the disease gene. If there are more markers in that region or they are closely linked with the disease, there will be more chances to identify the disease gene. Here is the example of the genetic mapping in case of humans. This is the chromosome 11, and a gene is present on P arm. This is P arm, this is Q arm of this chromosome, and a gene responsible for sickle cell anemia is present at a position 11p15.5. So this is the location of this gene. In this disease, amino acid that is polar in nature is converted with another amino acid that is valine, which is non-polar. Due to changing of this amino acid, the individuals which have this change in their normal sequence, in the normal sequence there will be adenine, while in case of affected individuals they have thymine in place of adenine. This is A is for normal adenine, and in place of adenine there is T-thymine. So adenine is replaced by thymine. The individuals they develop a disease. That disease is called as sickle cell anemia. Genetic mapping is a method that provides the clues that where are the genes located on the chromosomes.